This is a production of Cornell University.
Thank you very much for the generous introduction, Tim, and good afternoon, everyone. Can you hear me okay? So today I'm going to be talking about my part of my PhD thesis. I call this part one because I'm hoping that you will come back for my exit seminar, which will be part two. And we're going to be looking at contrasting environments. So by way of outline, I'm going to be talking about cassava. Hopefully by now, cassava is a very familiar crop because there's a lot of research ongoing at Cornell. I'll be talking about cassava flowering. I'll give you an overview of my thesis, and then I'll talk about the main results from my experiment, from the part of my work I'm sharing today, and then we'll summarize that. So first of all, cassava is a big food security crop, particularly in Africa, and also it's used as feeds, and this is in Thailand. They export most of this to Europe. And then because the tuber is almost 100% starch, you can see this in the background, it's almost 100% starch. It has a lot of industrial applications. So this is why cassava is a very important crop. But in addition to this, just a little bit about biology of cassava. Cassava is part of the Euphorbiaceae family, and it's a perennial shrub. The center of origin is in the Amazon, so that's South America. I showed you, I think this is just a fun slide of the numerous kind of things that we can get from cassava by way of food. So this is fufu, it's a fermented cassava. This is gari, fermented plus roasted cassava. And then you can also, some parts of Africa, the leaves are also used as a vegetable like spinach. Cassava grows in the tropical regions of the world, and so you see this distributed nicely, the latitude represented here. And a large proportion of that production takes place in sub-Saharan Africa, and this kind of gives you a reason why it's a huge food security crop. Traditionally, in sub-Saharan Africa, cassava is grown as insurance against total crop failure because it can survive on marginal soil. So if all else fails, cassava will survive. So you have it growing on the side in many farmers' plot. This is what the plant looks like on the field. I'm going to talk some more about the morphology as I progress in the slides. So why cassava flowers? The harvestable part of cassava is the tubers, so the flowers are not end goals as it were. The reason why we need flowers is because of breeding. So you can't, well, there are many ways to breed right now, but flowers are important instruments. So your breeding cycle or the success of your breeding program rises and falls on the availability or the success of flowers. So this is why flowers are important. So it's mainly because of breeding. Here are outlining some of the problems with cassava flowering. So as you go all through the development of the reproductive phases, you have different kind of problems. First of all, the flowering time. Some genotypes, particularly the ones the breeders would love to work with, is late. And then if you do go through eventually inducing flowers, development could be arrested prematurely. So an example here, way before anthesis occurs, the flowers are aborted. Well, if you succeed in the time and the development of flowers, the sex ratio would be another problem. There are 90% males compared to females, so we know that females are going to be the seeds that the breeders will work with. So if you succeed in this too, you're going to have a problem here when you don't have sufficient females to work with. So this is because cassava is an outcrossing species. Well, if you succeed through 1, 2, 3, you can also have problems in seed set. The fact that you have females to work with doesn't guarantee they will set seed. So the result of all of this is your breeding is suboptimal. Actually, you end up skewing your breeding to just the genotypes that are able to produce flowers you can work with. From our studies here in the Starter Lab at Cornell University, there are a number of things we've learned about cassava. I will give you details about each of these. We've learned definitively that cassava is a long day crop, so it's long days favor flowering time. We've also learned about a special type of branching that is associated with reproductive development. We've also learned about the temperature. It needs moderately cool temperature. A lot of my work focuses on this. And then we'll talk about we've also learned that the flourishing as in other species, flowering locus T is involved in regulation of flowering. So to describe what forking looks like in cassava, I have this model here. I'm going to walk you through it and show you some description of how flowering takes place. So first of all, on the y-axis, I have the percent of plants flowered. This is actually from data, just bunch of data. I made this up with this. So the number of days to the first flowering event could vary by genotype. So some could be really early, some could be very late. But after the first forking event or the first flowering event, you have a period of vegetative growth, and then it moves to the second and to the third. So I want you to keep this in mind. When I talk about tier 1, 2, or 3, I'm talking about flowering events, the first event, second event, and so on. By way of how the plant grows, this is what it looks like. So in this particular line, this is going to be an example of a good flowering line. So you have initial vegetative growth, and then you have this fork type branching and inflorescence in the middle. So this will be the first tier. Then you have a second tier, and then you can go on and so forth. Some genotypes don't have as good flowering. They only succeed in just having one flowering event. And some genotypes just never flower at all. Well, for us, this is why understanding the biology of cassava flowering is important, because in such genotypes, there are a lot of traits, maybe disease resistance or high yield or whatever. It's locked up in this kind of genotype, and you cannot include it in your breeding cycle because you don't have flowers. So this is one of the reasons why our study is important. Still giving you some details about the morphology. I talked about a special type of branching. I wanted to distinguish this from other types of branching that can occur in cassava. So you can have lateral branches, like so, but this is different from the fork type branching. So the fork type branching is the reproductive branching. With lateral branches, you can decapitate a plant, and the buds on the plant are activated, and that grows. But you can get this at any point in the growth of cassava, but this is different from the fork type branching. It only happens when the plant is transitioning from vegetative to reproductive phase. I told you the cassava is a long day plant from studies here in Ithaca, definitively, and this is some data to just support this. So you see in all across this table, only at long days do you have some flowering. At short days, you really wouldn't have any flowering, and the age to flower also corresponds with that. I told you also that flourishing FT is involved in flowering, and we can see this response when we grow the plant under long day condition. Particularly FT2 is responsive to the long day photo period. So this is where I begin my story little by little. Before me, a previous member of our lab, Sarah Deyemo, and also Peter Hyde here did a lot of work on temperature. There's more on this still ongoing, but I'm connecting some of my research with this. And you can see here that at cooler temperatures, you have a lot more flowering. And it's possible that at warmer temperatures, you have no flowering at all. You have slight improvement in the days to flowering, in the age at which flowering occurs when the temperature is cooler. So my PhD thesis, this is like a lot of information to work with, and like I kind of decided that I'm going to contribute to cassava flowering in 3 phases. I'm going to look at the abiotic environment, to look at the roles of hormones in this, and also study mechanical status, and hope that will give us some information on cassava flowering. For today, I'm only focusing on the abiotic environment. These other 2, I hope you come for my next seminar, and then we'll give you more information about them. Historically in Ibadan, there's been this story that there are 2 locations in Nigeria. This is Ibadan and Ubiaja. And the dogma is everything that you take to Ubiaja flowers. So breeding programs both from IIT and Ibadan, from Umudike, they come to Ubiaja to make their crosses, because for reasons which no one could say previously the Obiaja environment favors flowering. And this is some of the historical data I was able to lay my hands on. And you can see that Obiaja is earlier than Ibadan in terms of its flowering. So my job, my interest was to find out what the underlying biology in this genotype by environment interaction was, and also hopefully that will provide us some insight that we can begin to suggest ways to intervene and to improve cassava flowering. What I did was, so from IITA maintains a 700 genotype population called the genetic gain population, and I selected genotypes based on the data that they had in three categories. So genotypes that were between 30 and 60 days, I considered those as my early category. My mid category was 60 to 90 days, and my late category were genotypes that were over 90 days. Of course, I can't work with the 700 genotypes, so I made a sub-selection. I planted these simultaneously in both location, and I observed the physiology of the plants, and also was collecting data on the environment. The first thing is temperature, and from the data collected in 2017, what we learned is Ibadan consistently led Obiaja in temperature, so Ibadan was a warmer location. Notice in month 1 and at the 4th month, you have like this huge difference in the well, maybe not huge, but you have a difference in the temperature, maximum temperature. So I want you to bear that in mind. I was also able to collect rainfall data, and Ubiaja turns out to be wetter than Ibadan, possibly explaining why it's polar. I want you to remember that Nigeria is a tropical country, so day length is not a factor because day length doesn't change as much. So the only reason why it will be cooler is not because of snow, but it's because of rain. So from the physiology of the plant, looking at plant height as an index for plant growth in the two locations, indeed, the plants grew better in Ibadan compared to Ubiaja. And I just wanted to remind you that from the previous slides, we talked about the weather, and Ibadan is warmer, but it's not drought stress. So there's sufficient rainfall to show that it's not drought stress. And so it's possible that the growing conditions in Ibadan favors photosynthesis, so you have more biomass production, and like that is why plant growth is much more rigorous than in Ibadan. This pattern was also true for shoot biomass and root weight, but I'm not showing that here. However, we don't see any evidence for genotype by environment interaction because the same rank maintained, sort of the same rank maintained in Ibadan, except for a few genotypes is kind of maintained in Ubiaja. But in flowering time, the story is slightly different. So I used a number of nodes to forking because this is a kind of a developmental time score. In our Arabidopsis, you use number of rosette leaves to as an index for the age at which plants flower. So I use a number of nodes to forking, partly because these doesn't change. It's kind of determined. So however, whenever you come to score it, it's when it's determined, it's determined. So it was a much more objective trait to use than the chronological age of the plant. And we see the genotype by environment interaction being how it plays out here. For some it's strong, for some it's not so much. And all genotypes were earlier in Ubiaja than in Ibadan, supporting historical data that we had that Ubiaja genotypes are usually earlier in Ubiaja. But like I said, some of them, there was almost no difference, like they were almost on top of each other. So it was almost identical in the two locations. It turns out that these genotypes that behave this way happen to be the genotypes I had selected to be in my early category. Looking at this, I compared the two locations, and I was asking the question whether whether this change was statistically significant or not. And this placed the genotypes in two categories. A category which I will describe as plastic, and these genotypes were significantly different, so meaning the change and time to flowering in two location was significant, whereas those that I would describe as non-plastic, which the change was not as significant. I want to pause here to define plasticity just in the interest of those who may not be biologists as the response of living organism to the environment. And so the plastic genotype means that they are responding very strongly to the environment, while the non-plastic genotype are sort of maybe insensitive to the environment. The result I saw from my own study made me to go back to the historical data I had access to from Ibadan. So this was data collected in between 2013 and 2015, and I wanted to see how plasticity plays out. I didn't have notes to fork in because that was something I introduced when I started my own study, but instead I had the chronological ages of the plants. And what I did here was essentially to take the difference between the days to flowering of the mean days to flowering of the genotypes in Ibadan and Obiacha, and most of the genotypes were later in Ibadan. Some of them were stable and or non-plastic, while a few of them were the reverse direction. The non-stable line, I defined my threshold for stability as lines that had no more than 3 days difference in their flowering times between the two locations. So this is like something like a way for me to begin to think about the biology of what is going on in the two locations, not just one location is earlier than the other, but to see that there is this interaction with the environment, and each of the genotypes can behave differently. So zooming in more detail, I took a representative member of the plastic group and a representative member of the non-plastic group. And on your left hand, you have the data from number of nodes. But what I'm showing here is something a little more advanced beyond just the time to initial flower induction. We have here what happens over the course of 3 years. So for the first genotype, this genotype, which is the same as the non-plastic genotype, you see that the pattern of flowering is similar in the two locations. At the same, but in the plastic genotype, the pattern of flowering for the 3 tiers, so this is tier 1, 2, and 3. Remember, I described the forking event, so you have this different. At the same time, if you compare each of the genotypes in vertically, so like looking at each location separately, you see that it's Ubiaja, which is a favorable environment, has a similar pattern in the plastic and in the non-plastic and the plastic genotype. So I'm taking that to sort of reflect what is going on here, that there's a sort of a convergence or similarity between the two genotypes in Ubiaja. But if you look at the other direction, the unfavorable location, you can see a huge difference. If I should say more about this, moving the time to move from one flowering event or like one tier to the other in the unfavorable environment expands, and also the proportion of plants that move that are able to have subsequent reproduction, reproductive phases gets lower and eventually is completely shut down in the plastic genotype. So this was an interesting thing that I decided to dig a little more into the biology of this. But before this, from the genotypes I had on the field, I shipped a number of them to Ithaca so that we could do controlled studies on those. And with Peter's help, I was able to get this running. So this on the field, these are genotypes from the field. So I'll walk you through slowly. So you have the plastic, the blue and orange line represents plastic genotypes, those that respond to the environment, while the green line is representative non-plastic genotype. Under growth chamber or under control conditions, we had 3 temperatures, 22, 28, and 34 degrees daytime temperature. And you see well, this resembles the field a lot, that the plastic genotypes on the field were very, very temperature responsive. So meaning that as the temperature got higher, their days to flowering, the time to flowering also increased. Whereas the non-plastic genotype was almost temperature insensitive. Of course, I know you might say, oh, they didn't follow exactly the same pattern. The blue is not quite as steep as the orange in both locations, but it's just an idea to begin to like help us to think about what is happening on the field. So remember, the field is not just one. We cannot reduce field conditions to be just temperature, but this is kind of showing evidence that the thing going on the field might be temperature related. In addition to this, previously from our lab, we had also done a previous member of our lab, Sarah Adeyemo. She had overexpressed Arabidopsis FT in cassava, and we found out that improved flowering. So we took the two genotypes in question. So the wild type 60444, it's a South American line, and it's a late flowering genotype. Well, in this, I hope after this presentation, we would redefine what late means. So late would be in reference to a particular environment. And also the transgenic line, which carries the overexpression of FT. And just like before, the late or the wild type had a lot of plasticity. I want to draw your attention to the fact that this horizontal line here doesn't mean that it no longer responded to temperature. It's just because in the growth chamber we couldn't grow beyond 180 days, so we had to truncate the experiment. So that's why. So it's possible if we had enough space and kept it going, we could have seen this line go further than this. Whereas the overexpression line was sort of insensitive to the temperatures. So based on this information, I proposed two models of what is going on in cassava with respect to plasticity. My first model, I'm suggesting that in the non-plastic genotype, a lot depends on the genetics of the plant itself, while the environment contributes very little. My hypothesis is that in the non-plastic genotype, sufficient flourishing or flowering stimulus is produced such that the environment doesn't affect it much. So in the quantity of flourishing that it produces is able to counteract the anti-flourishing, so it's able to progress to develop flowering time independent of the environment. In Arabidopsis, there is the autonomous flowering pathway, and I can begin conversations about how this might connect to this. Whereas in the plastic genotype, flowering time depends heavily on the environment to facilitate the one of three things. The environment can either increase the flourishing, such as in a favorable environment, increase the flourishing so that it's able to counteract the anti-flourishing and then have flowering time. Whereas the unfavorable environment may not do that. You can also do the reverse. It might be that it's not just the flourishing, which is the flowering stimulus that is changing, but the suppressant of flowering, so that's the anti-flourishing. And also, we can also propose that the two are changing. So we provide a favorable environment, a lot of flourishing is produced and a small amount of anti-flourishing, but in an unfavorable environment, the reverse happens. So this is sort of what I'm proposing to that is potentially going on in the plastic and non-plastic genotype. But Sarah also in our lab had already done some studies on temperature, and she was previously I showed you that FT was the FT2 in particular was responsive to photoperiod. So at long days you had more FT production. So she was also interested to see how temperature affected this FT, and I want to connect what she did to some of what I'm finding. And in the stable line, we don't have any evidence that the temperature is affecting the FT expression in the correct direction, because at lower temperature you should expect higher FT expression compared to warmer temperatures. It's possible that I'm speculating that because this line is a non-plastic line, it doesn't depend on the environment, and so what the environment is doing to the FT expression may or may not have any bearing on the phenotype, which is flowering time. Whereas in the plastic genotype the FT1, there is no evidence for there's no evidence that this is temperature responsive, but FT2 shows some. Well, we can argue about how strong this evidence is, but at least there is some evidence that at cooler temperatures there are higher FT2 expression compared to warmer temperature. This is just the aspect we have for Florigen, but we don't have any information on the anti-Florigen. So I was interested in doing a more global study about using these two genotypes so that we could get some information on the anti-Florigen as well as the Florigen. So just quickly, many people know about RNA-seq, but RNA-seq just uses a whole genome expression profile and allows for quantification and identification of the whole transcript. We use the R3' RNA-seq method, which was cost-effective, and RNA is reverse transcribed to cDNA, and then we sequence the 3'. And I also want to remind you quickly that Ibadan was in February location, and earlier the temperatures in Ibadan is warmer than Obiaja. And this is what I did. I had two genotypes of plastic, representative plastic and non-plastic. I had an unfavorable location and favorable location. I took 2 time points. So one was my representative juvenile phase, 30 days after planting. And then I took 7 days after forking. So at the stage where I was sure that flowering or transition to reproductive phase had occurred. Extracted RNA in Nigeria, shipped it to Ithaca, did some more processing like purification and so on, and sequenced that biotech. With the help of Yihao and Hugh, I would say we got some of the statistics ongoing. And this is a double-centered PCA methodology, which Hugh can give more information about how that is done. But basically, on the main contrast is shown on the abscissa on the x-axis, and we see there's a clear sorting out between the two genotypes. In the blue genotype, we see that the open versus the closed is separated into 2 different segments of the PCA. Well, by the way, I should mention that the double-centered PCA works with the interactions, not just the individual elements. So the interaction of the samples and the genes and the expression values. And we see that there's a clear sorting. So meaning that in this genotype, just as we've seen in the phenotype, there's a clear sorting into clear distinct behavior of the location. So all of the Ibadans and all of the Ubiajas went into different components. Whereas the non-plastic genotype, you didn't have that clearly sorted. But what is seen is that the developmental stage is a stronger role in the non-plastic genotype, which makes sense because if you sample at 2 different ages, you expect plants to be different. Looking also at the fold change, I took the reproductive phase just as an example. There is more information about this that can come up later, but to illustrate what is going on just for the purpose of the seminar. At 7 days post-forking, which is when all plants had transitioned to reproductive phase, you can see that in the non-plastic genotype between the 2 locations, there's very little change in the expression or in the way the 2 locations behave, whereas in the plastic genotype, you have a lot of difference in signal. So these are things that we can even begin to dig in further to look at what is happening, what pathways are involved, and so on. So I'm going to summarize right now, and I am saying that on the field, the plasticity and the response, the genotype by environment interaction we see on the field is related to temperature, and particularly favorable flowering is related to cooler temperature. And We've also seen that at cooler temperatures, the flowering time converges irrespective of the genotype. In plastic genotype, warmer temperatures delay flowering. And when there's high expression of FT, we are able to reverse this plasticity. Our gene expression profile shows is able to distinguish the plastic from the non-plastic. And this is something I'm still going to look in more details, that warmer temperature might stimulate the expression of the anti-florigen, which might explain some of the plasticity which we're seeing. It's not the end of my PhD, so I told you that I'm doing today I'm focusing on the abiotic environment, and I'm going to do much more work on this in the days coming, and then I'm going to complete analysis and interpretation of data on the cytokinin and sugar status. I want to acknowledge the many people who have helped me there. This slide does not do justice to all the people who have helped me at the course of my journey. And I want to acknowledge my PhD committee, the members of our lab, both present and past. I had a lot of help from Yi Hao and Hugh on the RSC data analysis, my collaborators in Nigeria. I had a wonderful field team, particularly Yemisi, my field assistant, who has become more than just an assistant, who is much more like a friend. And a lot of undergrads helped me with my phenotypic scoring. Well, this doesn't capture all of them. I had several rounds of undergrads working with me. I had a small grant. Well, don't underestimate what small grants can do to your research. And then funding I've received from IITA, support by field spaces, NextGen through the Bill and Melinda Gates Foundation, and of course the government of Nigeria. So Eshegon is how we say thank you in my language, and I'm open to questions.
Are there any questions?
You indicated that the flowering, I mean, the evidence says that the flowering is better when it's cooler. Do you have any suppositions as to why it's better when it's cooler?
Yeah, so that's kind of the whole goal. The model I showed was it's possible that at cooler temperatures, there's more inductive signal being produced, and this is able to counteract the inhibitory signal. So that's essentially my argument.
From an evolutionary perspective, how would something like that have come about? What would you think the thing is that's in the environment that would encourage that type of?
Well,
that's stretching. I know, asking a lot, but.
Okay, so I would attempt, but it may not be satisfactory. So I'm looking at a tropical species here. One way you could think about it is that the cooler temperature is not favorable for the plants, and so it needs to accelerate reproduction so that its life is, well, it continues succeeding in the environment. So like when it senses that it's getting cooler, like let's quickly reproduce so that we can continue our lineage. But that's for tropical plants. In temperate species, you have mineralization, which is sensing when winter is going to end so that reproduction can take place. So that's the way I think about it, that it's everything a plant would do is to favor its reproduction. And so if it notices that something is going to affect its succession, it would quickly reproduce before.
So in a sense, it's a stress response. Kind of like we have mass years with acorns and things like that. So there's a bad year, and the year after they have an abundance of seed. So are you thinking it's something along that line?
Maybe not exactly. Maybe just being ready, because if you look at winter versus spring, it's just that the resources that are going to be available in spring is going to be abundant. So we need to flower so we can make the most of the resources available, not really that we are scared that we're going to die. So that's the way I think about it. Maybe not satisfactory.
So it's looking for an advantage rather than avoiding the problem.
Well, in our case, I have this thought that because it gets cooler when rain begins, my thought is also somewhere along the line that there's more abundance of water, and so the plants can distribute pollen more efficiently using water, for example, or growth is going to be improved and so on. So those are my thoughts.
You showed a plot of this differential between embodiment and yada, and I guess what I'm trying to think about what data we have in projects that would so all of these genotypes have been sequenced or been genotyped, all these individuals. So, but it's kind of detailed. Well, the days of flowering is not something that the readers routinely take, I guess.
So you probably know, since you're part of Next Gen, you probably knew there was a GWAS flowering time that was done at some point. So some of that data is
Okay.
got access to.
Did they? I should know this, I suppose, but did they look for loci that specifically interacted with location
I don't.
that were affected by this plasticity, stability, and things?
So the plasticity bit was kind of my idea about how the way to look at it. But I know from that study we had a lot of small effect QTLs, and it was really spread out so much that we cannot.
Okay.
Flowering is an environmental trait, like a trait that is. The QTLs we found, or they found, was possibly just environmental. So.
So I'd like to just say a little bit more about that. So there was a PhD candidate, University of Ghana, WACI program, and this was University of Oregon. And she examined, this was her work, 3 years of study about the age of the 700 genetic population, and she identified QTLs for flowering. I do not remember any effort to try to separate out or to look at TYE between the two locations. I also remind you, your postdoc, Martin Wolf, also worked with personnel on this project. That hasn't been published yet, so maybe we can see something along the lines.
Any more questions?
Well, here's a question.
Just a general question, Cassava, does flowering influence root yield?
Okay, so I actually have one experiment in which I looked at that study, and can I go? Yeah.
Well, I don't know if it's conclusive. Go ahead and tell us.
Okay.
What
Okay. I have my speculation based on my result is that it's sort of independent. I know you're thinking about potatoes because the florigin signal influences tuberization as well as flower induction. But remember that potatoes is a modified stem, whereas cassava is a true root. And also the phloem, the way the phloem is arranged in the stem is not quite the same as in the root. FT is produced and transported through the phloem. And so those are kind of where my thoughts are going. But my data kind of suggests this independent, but not conclusive.
Okay, well, if there are no more questions, let's thank Rick.
This has been a production of Cornell University, on the web at cornell.edu.